for the kind introduction. It's my great honor to present our work today at this prestigious conference. I declare no conflict of interest regarding the presentation. So the brain parenchyma contains approximately 80% of water, which is substantially higher compared to about 60% in the total body. This demonstrates that the brain is one of the most water-rich organs, highlighting the importance of water homeostasis in the central nervous system. To elucidate the dynamics of water molecules in the living brain, we utilize the stable oxygen isotope 17O labeled water H270O, as the former speaker explained, as an MRI contrast agent. And the 70O isotope adjacent to the proton causes vicinal coupling, leading the shortening of T2 relaxation, also T1 low relaxation. Because 3D conformation of H270O is almost identical to regular H2O, because the angle making by 2 proton and 1 oxygen is almost the same between these molecules. It can be used to detect alteration in water mobility or water dynamics in brain tissue sensitively. In H270O MRI, the magnetic resonance signal of 70O allows visualizing and quantifying water dynamics, similar to radioactive ligand using on a PET study. Accordingly, appropriately designed 70O-labeled molecules can be applied in the many of MRI study. In this research, using T2-weighted imaging, this can be developed into non-invasive imaging, namely JJVCPE imaging, that is capable of quantifying the content of the target of molecules, akin to radioactive tracer imaging such as PET. By continuously occurring T2-weighted MRI images before and after intravenous administration, just a moment. So to eliminate T2, effect of T2 star, we made a diabetic multi-TE fast spin echo sequence, and adjacent to TE equal 88 milliseconds. That is almost appropriate to measure the intravenous H270O. That's almost 2% in the brain in this research. To clarify the roles of specific water channels occurring in brain water dynamics, we compared AKP1 and AKP4 knockout mice with wild-type controls. AQP1 is strongly expressed in hyaloid plexus epithelium, whereas AQP4 is primarily water channel in astrocyte, especially in the Zeno and Hood process, and covering synapse throughout the brain parenchyma. On the left side of this slide, you can see the representative time course of signal intensities within pixel of each region of interest. The colors indicated different region: blue for the cortex, red for basal ganglia, and green for cerebrospinal fluid within the third ventricle. Each dot represents the intensity of each pixel within the ROI before and after intravenous H270 administration. The data shows that the movement of H270 in the brain parenchyma was significantly reduced in AQP4 knockout mice compared to controls, indicating impaired water dynamics in contrast, AQP1 knockout did not impact the brain H270 kinetics. Quantitative analysis confirmed that the partition coefficient of H270 between cerebrospinal fluid and brain parenchyma was selectively decreased in AQP4 knockout. This suggests that AQP4 critically contribute to water dynamics into the brain parenchyma and out of brain parenchyma to CSF. So story is a little bit changes. Amyloid beta peptides are continuously produced during neuronal activity, and their clearance from the brain parenchyma is central to the pathogenesis of late onset Alzheimer's disease. We hypothesized that amyloid beta is cleared through so-called glymphatic system. And impaired clearance of amyloid beta along the perivascular space due to reduced water dynamics could promote its aggregation. To test this hypothesis, we examined H270 dynamics in an Alzheimer's disease mouse model expressing mutant human amyloid precursor protein and presenilin-1, namely ABPPS1 mouse. So still presymptomatic and has not harvested aggregated amyloid beta, which is called senile plaque. At 17 months of age, these APP1, PS1 mice already exhibited showed parenchymal water mobility comparable to AQP4 knockout mouse. Positron emission tomography imaging using 15-level water was translated to the human study of water dynamics. Group suffered from late onset Alzheimer's disease showed lower perivascular flow and impaired water dynamics compared with both young and age-matched control with almost cognitive normal. But interestingly, age-matched cognitive normal group tended to split into two groups, namely cases with almost identical water dynamics compared with young normal group, and cases with decreasing water perivascular flow group. Intriguingly, a proportion of cognitively normal elderly subjects had having declining water dynamics compared to comparable with Alzheimer's disease patient. This red dot guys. 4 years clinical follow-up revealed that individual tended to harvest amyloid beta, aggregated amyloid beta, showed on full metamol 18F PET image. Integrating our findings, we hypothesized that improving brain water dynamics by facilitating AQP4 function could help suppress amyloid beta accumulation, thereby mitigating Alzheimer's disease pathogenesis. Initially, we chose potential compound with low docking energy to AQP4 in silico. We then assessed the effect of these compounds on regulating water permeability of AQP4 by examining the hypotonic expansion of oocyte of xenopath that made expressing AQP4 by cyclic DNA. Subsequently, through the JJVCP study conducted to mice, we identified the compound that capable of modulating the water permeability of AQP4 in the brain. First we created an AQP inhibitor, TGN-020, which has been used to 54 papers at this point. It has been effective in mapping human AQP4 as a PET tracer, as well as preventing ischemic brain edema in mouse and rat model of cerebral infarction. Then, we have also developed an AQP4 facilitator, TGN-073. I will briefly describe the effect of this compound on animal model of Alzheimer's disease. Gadolinium DCE MRI, as former speakers used for mice and rat study, shows that intravenous TGN-073 administration enhanced the brain penetration rate of MRI contrast agent in a wild type mouse, indicating facilitated water mobility. We also assessed the the effect of TGN-073 on amyloid beta clearance in hippocampal intestinal fluid using in vivo microdialysis in awake, freely moving Alzheimer model mice. TGN-073, this red line, significantly increased the elimination rate of 5xFAD Alzheimer transgenic mouse. Finally, oral TGN-073 administration for 3 months, from 4 months age to 7 months age, significantly mitigated the accumulation of aggregated amyloid beta in the hippocampus and cortex of 5xFAD transgenic mice. In summary, our integrated research elucidated brain water dynamics as a potential target against Alzheimer pathogenesis. Enhancing AQP4 function by TGN-073 could facilitate amyloid beta clearance, providing novel disease-modifying strategy for Alzheimer's disease. Thank you for your attention.